afternoon or this morning, wherever you are. I'm going to talk today about the Opal Multiplex staining method for tissue phenotyping. Opal's actually a subject fairly close to my heart. I've worked with it for probably the last almost a decade, really. But and I think it's kind of one of the most important things we've had happen in the area of histology for quite a long time, and indeed, it's opening up a whole new avenue for tissue applications. So what I want to do today, if I can, is just to go over a number of points, which I hope you will find interesting. The talk is kind of directed towards more towards you who have not really done Opal much in the past. So for those who are really expert in it and are listening, hopefully it might be a bit of a revision. I'm going to talk about the multiplexing, a little bit of an overview, a little bit of Opal in detail, a little bit about the chemistry and how it works. As I think in any application we do in the lab, it's good to know what goes on behind it. Then some optimization information. And then finally, towards the end, talk a little bit about the imaging that's related to this multiplexing. Okay, I'm sure you'll all recognize that. It's a hematoxin eosin section. And you recognize it because you understand, or a lot of you will understand, the tissue makeup. So histology generally is a very descriptive and morphologically based tool, and has been for probably the last 150 years, and it forms really an important backbone to general pathology and tissue analysis. But it really only gives you the overall architectural relationships between groups of tissues, the groups of cell groups within a tissue. So here we might have, in this case, as a breast, we might see gland structure, we might see some adipose tissue there, we might see some connective tissue down here, we'll see a little bit of inflammation down here. So, however, it doesn't, besides giving us a morphological view, it doesn't really give us a lot of information into what is actually happening on a cell per cell basis. And this is becoming more important as we move more and more into the area of precision medicine. Now, we do have ways of trying to get more information at a cellular basis from histopath sections. And this is really through traditional techniques like immunohistochemistry, maybe special stains. These are down the slide. An experienced user, a person who's trained in tissue pathology or tissue analysis can look at it, and they will give some sort of evaluation, usually, particularly if it's a pathology sample, maybe a tumor. They will give it a rating based on their observation, based on their experience, to see what the degree of the tumor may be. So this is called scoring, and it forms a backbone of a lot of diagnostic areas in pathology in the past. However, as good as this application may be, as good as it is, and as effective as it is today still, it is basically largely based on experience, on your learned experience. It can be quite subjective. It's at best qualitative. It often shows poor reproducibility, high variability in between samples, statistically questionable, and not really up to the demands, I guess, of what we're looking for as we go forward into, particularly into precision medicine. And most of all, of course, it's very time-consuming, both for the people who have to create the sample and those, of course, who have to look at it and report on it. So as we move into precision medicine, the needs for change. We need to change such that we want to take the information that we get from non-spatial information about the cell, that could be from mass spectrometry, flow cytometry, new gen sequencing, whatever, and to be able to apply that information specifically to different cell types that we might find in the tissue. So we need to understand, identify these cell types, and also get some sort of understanding of their position in the tissue. And this we can call spatial phenotyping. The importance of this is, as again we move towards precision medicine, we want to be able to use this information to allow us to get a better understanding or stratification of patients, so that we can better so they can get a better response to some of the therapies that are available, a more precise prescription in terms of the actual needs of the therapy for the disease they have. So the importance of spatial information, particularly down to the single cell, is really quite important now. And this is why we're finding that techniques like multiplexing are really quite important in this pursuit. Okay, in any tissue microenvironment, of course, we need to understand the actual tissue components, whether they be epithelium, whether they be necrotic, whether they be stem cells, or whatever. And these we can identify from the broader view of the section. But we also need to understand a little bit about the immune cell population as well of the tissue, particularly if we're looking in the immuno-oncology area. So then we need to identify T cells, B cells, myeloid cells, dendritic cells. We need also to be able to look at how those particular individual cells are going to react with tumor cells, and also how they're going to react with each other. Now, we do have a tool for this, and this is immunohistochemistry, and which has been in use for quite a long time. But as a tool for spatial phenotyping, it's reasonably difficult. We either use a direct immunolabeling method, or better still, we'll use an indirect immunolabeling method. But generally, we need to characterize the different biomarkers based on a panel of slides. So in this particular case, we have 6 biomarkers, which generally require 6 sections to be cut, 6 slides to be stained. Now, the pathologist then, or the observer, has to bring the information from each of those slides, coordinate it together so they get a better understanding of the whole overall view. It would be much better, in fact, to be able to have all of those stains on the one slide. And by combining all of these together, this is where we come up with multiplexing. The advantages of it is that then we can actually see the location of the biomarkers on a single cell, on the same signal cell, where in this kind of format, each section is from a different level within the tissue block. So there's no guarantee that what you're seeing and trying to place as to kind of associate with a single cell are necessarily single cells. So the need for having a single slice with all the dyes on it is actually a good approach. Now, to do that, to be able to identify all of these different probes on the one section, we need to find a method to do that, a modality to do that. And fluorescence is the best one of choice. A fluorescence molecule has a very specific spectral signature. So allowing us to label a specific protein, for instance, with a specific fluorochrome, allows us to identify that particular fluorochrome amongst the mass of other fluorochromes. So fluorescence itself then is an ideal cellular phenotype method because it can both visualize and, more importantly, fluorescence can be quantified in the same tissue section. So it then allows us to really look at and understand the relationship and distribution of cells within tumors and more importantly, the tumor microenvironment. Now, multiplexing immunohistochemistry doesn't come without its challenges. And this is one of the challenges, in fact, one of the reasons why fluorescence as a technique in histology really hasn't really been that popular. And these reasons can be that if you're trying to do multi-staining on the section, you have to worry about antibody species cross-reactivity with each other, which reduces their selectivity. You will have to watch for other conditions as well, and some of the more major ones are maybe single crosstalk between multiple dyes, that is the bleed-through crosstalk, background staining from the general sample, and very importantly, because we mostly fix the tissue in an aldehyde fixative, aldehyde fixation increases the autofluorescence within tissue structures and can increase it to such an extent that it overpowers the actual signal from the probe that you're looking for. So tissue autofluorescence is a major issue when we come to fluorescence. So in comes Opal. So what is Opal? So Opal is a fluorescent-based immunohistochemical methodology, which can allow up to 9 colors on a single section. Importantly, it can be used with any primary antibody source available, and they can be all from the same species. It's an accessible method to anyone who has some skill in standard immunohistochemistry. The dyes themselves are spectrally spaced for precise spectral unmixing, which is a technique that we use to interpret and to analyze multiplexed data. They are quantitative, and they can reduce the cost of your primary antibodies, as we'll see a little later. And importantly, they can be applied both to automatic staining devices or manual staining methods. Results you get from Opal, all on a single slide, can be shown in this set. So we can have the DAPI, we can overlay that with the tumor marker, we can overlay that with the cytotoxic T cells, we can overlap that with the regulatory T cells, we can overlap that with the B cells, macrophages, and PD-1, and PD-L1, and then proliferation marker Ki-67. And as you can see, the sample is the same, and now we've been able to relocate each of the individual cell populations within that tissue. So with the approach that we have, we can reliably image 9 colors and importantly, remove the autofluorescence. So Opal then is a fluorochrome, it's a fluorophore that we use for multiplexing. So its ability is to retain the spatial context of the tissue and the cells. It's quite easy to use. It's multiplexing this is quite straightforward to use, and it has the potential of unlocking even greater multiplexing and extracting a lot more data from the tissue purely than just the observational information, as we use the downstream processes of analysis on it. Now, Akoya supply Opal in a number of different ways. So we can supply it as kits or as components. So we can have a 4-color kit. Now, a 4-color kit has been designed for your standard fluorescence microscope with its general maybe 4 fluorescent filters on board. So this allows us to get good spectral separation, but still allows us to do quite easy multiplex on for the standard microscope system. If you have more automated instruments or spectral instruments such as the Mantra or the Vectra or narrowband filter systems, then we can move to an Opal 7-color kit. And finally, for the most flexibility and the highest plexing, we can go to 9 Opal dyes. But for this, you will need spectral unmixing, usually with a spectrometer. In this case, we use the liquid crystal tunable spectrometer, and this would allow you to be able to differentiate each of those dyes. All of them are for human, but we do have a mouse kit as well. As well as those kits, there is also predetermined tumor kits. So there's one for pan-canceromic kit and melanoma kit. Now, these kits come with all the required antibodies that you would need, as well as all the dyes that you need, as well as all the associated solutions to carry out the experiments. Okay, so let's now turn a little bit to the chemistry and workflow that you might need to do in Opal experiment. But before I go to that place, I would recommend that you go to Akoya's website and download this document called the Phenoptics Assay Development Guide. This is really a very, very useful guide if you're going to undertake Opal staining for the first time, or even if you've been doing it for a while. It details step by step the process that you need to do. It explains why you're doing them. And it really is the accumulated experience of Opal from many contributors. Opal's been in use now for quite a few years, so a lot of experience has been gained, and this has been summed up in this book. So I highly recommend that you get this book. It's free of charge, don't have to pay for it, doesn't cost a thing. You can download it as you like. Okay, so Opal is a multiplex immunohistochemistry technique, and it follows more or less the same setup that you do with standard immunohistochemistry. There's a slide prep start position. We have to put the tissue through an epitope retrieval process, which we'll talk about briefly shortly. We will then go in and use a blocking step to help to reduce background. On goes the primary antibody incubation, as the primary antibody unlabeled. Then we attach the secondary antibody, which is being conjugated to horseradish peroxidase, similar to what you might use in a DAB experiment. And then we apply the signal amplification step, which is the opal dye itself. After which we then carry out antibody stripping, which is applying the slide to heat immersed in epitope retrieval solution to actually break loose the antibody bonding onto the slide. This leaves the slide clean, except for residual leftover dye from the step, which we'll explain in a minute how that works, and then we'll start the cycle again. So the cycle repeats as many times as you need to apply as many of the dyes that you want. Then finally, it's at the final solution, it's then stripped the last time and stained in DAPI and mounted onto a slide cover glass. Look at that a little bit more in a kind of more descriptive way. Here's a slide which shows a piece of tissue with 5 antigenic sites. If we take CD20, for instance, and place it on that site, the primary, we can then label it with the secondary, which has the HRP attached. We then can stain it with the TSA Opal 520 in this case. And what happens is that the dye will accumulate around the junction between the primary attachment to the epitope and bind there. We then use a microwave source or a heat source to remove the secondary primary complex, and then we can start the process over again, step by step, till we finally get all the different colors that we need on our tissue. Okay, so the engine behind this is a chemical called tyramide, and it's part of a technique called tyramide signal amplification, or TSA for short. Been around for quite a long time, in the mid-90s. It's been around for quite a time. It's part of a technique called CARD, which is catalyzed reported deposition method, which is a group of methods which we can use to amplify the fluorescence or detection signal in immunohistology. Normally, if we just do standard detection, we might have one fluorescence molecule attached to the secondary, and we have to image that. To increase the imaging effectiveness, we can use the TSA to attach a large number of fluorochrome to the same site, and this gives a form of amplification. It can be used across almost any immunoassay technique, and not only light microscopy, but it's also used for electron microscopy as well, and when you're doing immunogold labeling. It allows us to give reduced antibody consumption, because now we'll get much more sensitivity from our antibody amount, so we can have higher dilutions and therefore still get a good signal. It has very good background resolution, and it can improve the sensitivity at least 10 to 100. I've even seen people quote that it goes to 1,000 times, maybe a bit hard to believe, I don't know, but I wouldn't doubt it. But it has many, many publications, well, many tens of thousands of references looking to that. So it's a well-established technique. In terms of opal dye is the binder that we put onto TSA for the opal work. So how does it work? So we find our primary antibody attaches to an epitope on the site of interest. Now, the chemical bond between these two is a hydrogen bond. It's actually a very weak bond, van der Waals forces only, so quite weak. We then attach the secondary, and the secondary has horseradish peroxidase attached to it. We then attach the TSA molecule, or introduce the TSA and opal molecule conjugate. These are then there's a reaction that occurs catalyzed by the presence of H2O2, which produces free radicals, highly reactive free radicals. Now, these free radicals will bind to protein structures within the material. Now, the two factors that are very important is it's very time dependent. These reactive sites only exist for a short period of time, and it's very spatially dominated. They only can occur when they're very close to the reaction area of the horseradish peroxidase. So what it does then, it plates out the primary and secondary, and of course around the horseradish peroxidase, but it will also bind particularly to tyrosine molecules that might exist in close proximity to where the primary antibody binds to the protein of interest. The binding of tyramide to the protein is not a van der Waals force reaction. It's actually a covalent bond. So it's significantly stronger in attachment than the weak van der Waals. So when we apply energy through heat, maybe it's a microwave to cause molecular vibration, or maybe it's through some other heating method, then the very weak van der Waals forces will break, and then we can wash away the complex of primary and secondary. What is left behind is the covalently bound molecules of fluorochrome and tyramide to near the site of interest. This allows us then to apply a second antibody, a secondary, and a second dityramide entity, and we can go through the process as you can see. The workflow itself follows very much a normal immunohistochemistry method. So slide prep, epitope retrieval, blocking, primary introduction, incubation could be for an hour at room temperature. For harder way, it could be 4 hours overnight, 4 degrees overnight, depending on what your antibody is. Introduction of your Opal secondary, your amplification with your Opal dye, and then the antibody stripping. This is repeated a number of times. And finally, then if you're not using the dye Opal Polaris 780, a very far red dye, then it would be stained in DAPI, and then mounted and cover glass. I should say the DAPI has to be separate, not a DAPI that's mixed with the mounting media. If you are using 780, this requires a different type of process to attach the final dye, and this would be done through an Opal TSA digitation bridge, and then we would go through the stripping process. Now, as you can see, if we were doing a large number of cycles and we had a large incubation time of each of them manually, we can spend quite a lot of time needed to do a multiplex. The good news is that the application can be very easily adapted to automatic staining machines. So both the Leica and the Roche are the main two that we'll probably see, although other systems can also do it as well. The advantage of these machines is that you can shorten the workflow from 3 full days to 1 evening. They're very flexible in the way you set them up, and very importantly, they're highly consistent compared to a manual staining method. So if you have access to staining machines, then this would definitely be the way to go. Particularly if you want to do more complex staining, multiplex staining, say in this case where you're using both Opal and RNA scope for multiplex fish imaging, then using an automated process, using an automated protocol, then this would make your life very, very much easier. So this would be something that would be highly suited to automation over a manual method. Okay, so now let's turn our attention a little bit to some of the things that you may need to look at if you're carrying out your Opal in the lab, and what we need to optimize, keeping in mind that it is largely an immunohistochemistry technique. Now, in the Opal optimization workflow, we need to look at a number of different stages. The first part is to calculate or to formulate our best primary antibody titration. So in that, we need to select the antibodies we're going to use. We would try to pair them with the opal fluoroforms. We'll talk a little bit about that later. We would look at, evaluate the staining pattern to make sure that it corresponds from opal to corresponds to maybe more standardized techniques such as DAB. And we would also try to optimize the concentration so that when we come to the imaging device, that the auto exposure times are all within a certain level range or levels. We can also use the optimized slide for a library slide, which is the second stage. Now, earlier, not so much now, but in earlier days, it was very important to create library slides because a library slide would allow you to establish the spectral makeup of the dye that we're using. So when you're doing spectral unmixing, you need to unmix a mixed group of dyes with a known and understood spectral signature. So what we would do is to look at a dye and measure the spectral characteristics of that dye as it's attached to the tissue, because the tissue itself can actually change the spectral characteristics of a dye. So it's important to have it within the same realm as what you're going to be using it on. And with that, then we can use that library to form a standard to which to understand the localization and the arrangement of the dye, the multiplex dyes on the sample. These days, a lot of this is done within the software itself, so the need for library development is not quite as important as it was in the past. Once we have developed good and satisfactory monoplex or single stain slides, we can blend together the processes that we've done into a multiplex. So basically bring the technique, multiply it from the number of cycles that we need to do. So we need to select the order of detection and finally do the multiplex, finally with DAPI at the end, and then we'll look for crosstalk and we'll look for balance. Finally, the result will be imaged, and then downstream, of course, will allow us to do our tissue segmentation, cell segmentation, phenotyping of the cells and measuring their expression levels, and finally validation. One word here, don't forget your essential controls. There's an immunohistochemistry technique, so it needs controls to make sure that what you're seeing is real. It's important things to keep in mind with optical optimization is horseradish peroxidase is necessary to drive this reaction. So that means that you need to be careful, you need to be aware of endogenous peroxidases that may exist in your sample, and they should be quenched to prevent non-specific detection. Now, one of the advantages of using a microwave, if you are going to use that technique for stripping and antigen retrieval, then the microwave itself does a great job in helping to reduce endogenous peroxidases. If the concentration of horseradish peroxidase is too high, those are the secondary that you're using is too high, then you'll get the formation of too many dimers, and these will actually lead to somewhat to reduce signal staining and a bit of a higher background. So because they've got more protein around to collect because of the higher HRP concentration. So for this reason, it's critical to optimize your dilution for your primary and your secondary HRP. And the use of blocking agents is highly recommended. When you are using your opal solutions, then we tend to standardize around about a 1 in 100 dilution. This just tends to work quite well. If it's a little too bright, we can reduce it. If it's a little bit too weak, we can increase it. But 1 in 100 is the target we're aiming for. Now, there's many factors that influence immunohistochemistry generally, and these also affect your opal staining as well. Not all of them are that important. Some you don't have control over quite often. Tissue handling, for instance, you're getting a biopsy material or from the operating theatre, then maybe this is not something you can handle, but something that can make a big difference in the result. But you can handle some things. Tissue fixation, epitope retrieval, and detection systems are things that in opal that you can certainly consider. So key components in any immunohistochemistry assay is really about the retrieval buffer, that is your antigen retrieval buffers, the antibody dilutants that you use, and whatever inhibitors or blockers that you're going to use. In the case of antigen retrieval, there's usually two types that we generally use. We can use a citrate-based pH 6 buffer, or we can use a tris-EDTA pH 9 buffer. And what is used is really kind of dependent upon what the vendor of the antibody will recommend, although pH 6 generally is quite a kind of a fairly close to a universal one in most cases. Antibody dilutants are the same as in the retrieval buffers. Quite often we work with what the vendor suggests for that particular antibody, and we will formulate using that particular advice. For blockers, H2O2, hydrogen peroxide, is a good step to use to try to quench endogenous peroxidase activity if you have a significant amount. And of course, keeping in mind that some tissues are much worse with endogenous peroxidase than other tissues. Took a slice of my skin, which is quite old. I'm sure you'd find a hell of a lot of it in there. So it does depend upon where your tissue comes from. High iconic strength or high protein concentrated blocking agents also can be used to decrease the blocking of the background, and highly recommended even for Opal. Now with Opal, we did a little bit more on the slide preparation, because what we're going to do is have to put our slide through a number of cycles. So therefore, we want to make sure that the tissue that we place on the slide actually survives the number of cycles that we do. Most of the time it's fine, but we take precautions to make sure it does happen. So always use good quality slides. Maybe Super Frost is one that we've used. And because these have this good adhesion properties on the slide. We always recommend that we bake before deparaffinization. We bake the slide 55 to 60 degrees, and this helps also the tissue to attach. Again, a fixation step, which is not always used, is a good practice as well. So fixation for 20 minutes in neutral buffered formalin also helps to stick the tissue. Now, some tissues obviously are more sensitive to floating than others. So it's not equivalent across the whole tissue band. Now, for those of you who want to look at using a microwave as an antigen retrieval method or stripping method, then just a couple of points that we've found from experience for this. So we find that optimal microwave treatment really does remove most of the endogenous peroxidase. It's quite amazing at doing that. So that's one of the good advantages of it. The microwave that we would recommend for Opal is a domestic microwave. Now, we find that a minimum of 1,000 watt is quite good. I've tried microwaves with much lesser power. They tend not to work as effectively, even though you do the same process as well as an 1,000 watt microwave. You need an inverter system. That is, that the microwave power is regulated by the power rather than by a timing interval, which is an older style microwave. It has a carousel inside and ideally has a number of power levels. Now, the Panasonic is a brand that actually does reach all those criteria. And so this is a good choice in microwave if you don't have one. For Opal, the microwave technique includes bringing the sample to boiling very quickly within approximately 60 seconds at 1,000 watt. And then after it's boiling, we reduce the power to about 20% to a simmer, if you like, rolling bubble. And then we do that for about 15 minutes and then remove and let to cool to room temperature. Usually we'll find the 15-minute time is enough to remove nearly or pretty much all the endogenous peroxidases. But if you have still have issues, then maybe you can do time series and staining and evaluate the point where your endogenous peroxidase disappears. A good sample for this is probably colon, because it has quite a lot of endogenous peroxidases inside. So if you can remove most of it from colon, you will certainly be able to move it from most tissues. Primary antibody titration is a major step. to find the right concentration for the Opal application. So usually we base this on the staining patterns we might see in a standard DAB result. So it's important to look at your standard DAB result to kind of mimic, if you like, in Opal the same response. We look at the signal to background as well. But with our starting point is usually the recommended dilution from the vendor. And then we will do further titrations, maybe 1 in 2, 1 in 4, in the 2 types of antigen retrieval buffer to see if each should make a difference. And then finally, if there's no difference, we might end up with choosing a 1 in 2 dilution at AR6. To visualize, we'll tend to use the Opal dye, TSA Opal dye, probably at about 1 in 100 dilution, as we mentioned before. But if you're using the machines, 150 for the bond, 1 in 50 for the ultra. What we're looking for is precise signet ring type structures. So in this case, we can see that it's stained, but if you look carefully, you can see that it's a bit, little bit spotty, a little. It's got little deposits, which means that all the sites for that particular antibody haven't been occupied. So with a higher concentration, we can start to fill it in a bit more, and finally we can get quite a nice signet ring structure. So hole in the middle, nice little, like a donut on the outside. That's what we're after, signet ring. Signal intensity is really about ultimately trying to finally, when you come to do your final multiplex, is getting a good signal for each of your colors. So we have a standard which we aim for. Now, this is calculated by using the Inform software that Akoya provides with the instruments that it has, the Mantra or with the Polaris or Vectra. And what we're aiming for is a count of around 20 to 30. Now, usually the 1 in 100 dilution will probably give you somewhere around that. If your antibody, well, the antibody concentration is the correct amount, then this is what we're aiming for. Okay, we want a good signal to background ratio, ideally about 1 to 10, so that this good signal to noise coming up. Okay, now some of the dyes are not going to always reach that. For instance, if you use Polaris 780, which is the deep red dye, it does have a very low intensity. So its number will be somewhere between 1 and 10, and that's acceptable. Some of the others are a little bit higher. So these ones, probably your antibodies can be certainly a little bit more dilute than maybe what is always recommended. Stripping efficiency, one of the important points is, of course, you want to make sure that when you do your stripping, that the antibody is actually removed entirely from the sample. So the new antibody that you place on is not going to be contaminated with the previous one. So stripping controls are important to do as well. So basically you complete a sequence, you would strip it, apply the secondary sequence without the primary antibody, but with the secondary and the TSA Opal, and then look at the result that you get. If you can still see some of the residual left over from the previous one there, then that means the stripping hasn't been enough. In which case then you will turn back and say, okay, look at my stripping time. Maybe I need to have a longer time for stripping. Or if I'm using a heat-based antigen retrieval method, then maybe increase the temperature from 98 to 100. This can be quite significant. This can be quite relevant to the staining machines. Okay, we're applying a lot of heat. Sometimes we're applying a lot of cycles. How does that affect our result? Does it really, cause a change in the way that the antibodies can react? Well, in actual fact, it causes little damage to the tissue. So cycled heat, induced epitope retrieval, HAER for short, is really not damaging to tissue. In fact, it can be quite helpful for your antibody staining. Some antibodies, some sites need more cycles to reveal themselves, to retrieve the activity from them. So in this case, for instance, Chi67 based on 520, we can see that after 6 cycles, we see an increase in the cell, in the intensity count. So this particular site has improved with that. Now that figures because sometimes the formalin binding of the site, which is the formalin cross reaction, which is causing the closing of these sites, can bind to different strength and different proteins. So therefore, by using a longer antigen retrieval methods, we can reduce strip the binding from the site and therefore make the site reactive again. Nice thing about formalin is it keeps the site, antigenic sites, mostly reactive once it's removed. Okay, are opal dyes stable? Well, yes, they are. Even after many, many cycles, you'll find that the intensity of the dyes remains constant. So this means that you don't have to worry about dyes towards the end of your cycles showing lesser sensitivity than the ones at the beginning. So deciding then the opal order, okay, which dyes go with which? Well, then it depends upon the expression level. Signal to background, do I need a stronger signal, do I need a weaker signal, and basically co-localization of my markers. So in opal pairing, we basically will have a series of opal dyes which have difference in intensity. So opal dyes like 480 are very strong emitters, where opal 780 is a very low emitter. So it would follow that the weaker emitters that we have would be better matched with the higher emitters of the dye, and the lower emitters, the higher emitters that we have from the antibodies, higher expressors, I should say, would be placed onto the lower emitters. So therefore, strong antibodies going on to strong sites going on to the lower signals of the 780. And this is so in this case, often the pan-CK, which is the cancer indicator, is a very strong emitter. So therefore, we often couple that with 780. Getting that correct means that you can co-localize some staining reactions even within a single cell, so that you don't get one dye necessarily overshadowing another one. The balance of the colors are actually quite good. So in a monoplex slide optimization, each biomarker should be optimized as a single slide. Should always include an unstained section for autofluorescence determination. And you should try to do your background and your primary and your sample so that your final counts are roughly 10 to 30. And this will end up then allowing you to get a good multiplex. In the multiplex slide itself, it's just bringing the monoplex protocols together. But you need to look out for signal balance to make sure that there's no dye overshadowing another. It should always be within intensity values about a factor of 3 between each other. The signal interference between them should be controlled. If one dye is particularly strong against another weak dye, what you'll find is you'll get black holes because the dye is covering that kind of called umbrella. It's covering the emission of one dye with another. And a crosstalk, of course, really, if you've got excessive crosstalk, then that means that you have inadequate stripping, and you'd need to go back and look at that. Balance is important because it influences crosstalk. So in this case, if we had a pan-CK with very high emission, there's always about 1 to 3 crosstalk of that dye into the 690 channel. If our 690, on the other hand, is very weak, then this high crosstalk will actually, from the pan-CK, will actually give quite a high crosstalk reading in the 690 signal. Ultimately, if it's balanced, the 650 and the 690 is similar, we'll still have the same percentage of crosstalk, but the contribution to the signal is far, far less. Now, the opal staining itself is really only a part of a whole process. So the staining is the first part of the Phenoptics workflow. It's then followed by the imaging in maybe the Vectra or the PhenoImager. There's image storage facility, a cloud-based image storage for distribution, and finally image analysis packages, both internally and external, for analysis of the data that you get. And it's because it comes the final result, the actual analysis of the data. So just a quick word on to finish up on opal and imaging. Now, here are the spectra of opal multiplex imaging using 9 images. Now, we can see that They're quite nicely spectrally spaced, but you can see a problem, I'm sure, and that is that they all have a very common base. They all overlap. So crosstalk, even with narrowband filters, can be a real problem when you're certainly looking at 9 colors. So the results are not quantifiable without spectral unmixing. And this is the function of instruments such as Mantra or Vectra or the PhenoImager as we have now, or Polaris as its old name was, because these have the capacity to do spectral unmixing. And this means that when we have a large group of dyes together, the filters are unable to really separate those who are closely matched. But if we know the characteristics of those dyes that are closely matched, we can spectrally unmix them. And we can do this either through software to some extent, but we also need hardware, that is through a spectrometer, imaging spectrometer, to be able to separate out the fluorescent signal. If we don't spectrally unmix, we are introducing errors into our phenotyping. So, because what we're doing is calculating not only just the pure dye that we're interested in, but also the contribution from the cross-match, the cross-talking dyes. So in this case, as you can see, if we take a sample with the raw data and then we have the unmixed data, you can see it's quite a significant difference in the result in terms of the numbers of cells that we get. Now, Akoya itself will offer quite a comprehensive framework for spatial applications. It's a spatial application company. We've talked about today just the phenotyping part at the Opal level, but there's also applications and for rare cell discovery where we're looking at very small amounts of cell activity within a whole slide. This can be done. We're looking, they also provide a spatial functional state. So we would combine the spatial biology with multi-omic and RNA and protein mapping to give more data about the cell position, within the tissue. There's a neighborhood spatial mapping, which needs to understand how different cells are interacting with other cells. And then we can also look at spatial signatures, which is has a strong clinical overtones, particularly through the high content study. So all of these are applications we haven't talked about. We only talked about the first one here, and I'm sure there's many, many webinars could be done on each of these. I want to also introduce a new concept from Akoya, which is the introduction of the phenocycler and what we call the phenocycler imager. Now, the phenocycler, unlike the Opal technique, is a fluidics-based system which allows us to look at around about 40 biomarkers at the same time. So this is a great device and a very important device for unbiased biomarker discovery. And this is linked to the PhenoImager, which can analyze whole slide using these large number of biomarkers, and it can do it quite fast. The results that we get from that can be then refined, maybe to apply to a more clinical setting through the PhenoImager and using the Opal staining method. So this is a new workflow connection from large number of biomarker assessment, very quickly assessed, and then move to a more clinical environment. The PhenoCycler Fusion is quite rapid. It does these within about 24 hours, a slide within 24 hours. So it's very fast for very, very large amounts of biomarker identification. You can look at both fresh section, frozen and formalin fixed, or you can look at archive material, which means you can go back and look at old patient blocks. Okay, looks at single cell or subcell resolution, and it's got quite high throughput for the option for the application that it has. So now Akoya really brings in the world's first integrated spatial biology system. So it really has expanded even from the early days of just simple things like the Opal technique, which was available long ago, it's now developed that technology into a very sophisticated biomarker system. So with that, the take-home message is to remember the importance of your tissue prep, optimize effective immunohistochemistry with your monoplex slides, always include an unstained section for autofluorescence determination, consider the order, and always aim for balance. So all I can say is all the best with your Opal multiplexing, and with any application, including immunohistochemistry, be diligent, and you definitely will work in the end. So I'd just like to acknowledge a few sources from where I put the slides together today, many from Akoya and a few from PerkinElmer. And if you have any questions, particularly go to the Akoya Bio website, as have a very, very good setup for large number of technical information, as well as articles and so forth. So this should be your first pearl to call if you are in fact undertaking any of these applications. So with that, thank you very much for listening. I think I'm pretty much on time, so that's always good. I tend to waffle on a little sometimes. So thank you for your patience and for listening. Thank you very much.
Thank you, Chris. Here we got some questions from audience. The first one is regarding our incubation time. The audience mentioned because for some manual methods, maybe some antibody need overnight incubation, but automatic system does not allow to do that. Could you please share experience about the transition from manual staining to automated system?
Right, yeah, so that's true. Some antibodies will need overnight incubation, maybe at 4 degrees. Now, that probably, if you definitely need to have that length of time, that probably is necessary. Although I think under the automated conditions, you'll probably find you can actually shorten that time a lot. So it depends upon the antibody itself. I won't say that the machine is going to take that away, and sometimes you might just have to resort to a manual method if that really is critical. But where the machine does take time is if you do have to, if the antibody is if there's storage needs to be done where you're doing, say, some degree of antibody incubation, but you can't complete the whole cycle in one day, then you usually need to store the whole system's antigen retrieval solution, in the fridge overnight before you can start the next one. So that's where you do save a lot of time, is where you maybe can't complete the whole cycle. But if you do need an antibody that does take a long time to actually work, then you may well have to resort to a manual method for that, I think.
Okay, thank you. Another question is, which chemical bond of epitope does the fluorescence bind?
Sorry, can you say again?
They say, what area or which chemical bond of epitope does the fluorescence bind?
Oh, okay, yeah, right. So the fluorescence part will bind to looking for just protein structures that are very, very close. It's more thyroid. What is it? Let me just go back and have a look. Okay. Terrible memory. Go back here. I see this one. Right, tyrosine is usually the site that it likes to bind. So it's going to bind to that molecule, which is a protein structure or in part of protein, and it's going to bind extremely close to this area here, to the junction of where the primary attaches to the actual epitope protein structure. So because it's needs to be influenced by the presence of the horseradish peroxidase. So tyrosine molecules which exist close to the site of the junction of the primary to the epitope is where the fluorochrome will bind. Now, it's very limited in its distance. There's been studies done, particularly with electron microscopy, where they'll find it's only a few nanometers, basically, away from the site of binding.
Okay. Another one is about the mixing. Should the customer use the in-house spectral library or the informed building library for mixing?
Okay, yeah, good question. Personally, I always like to use the library that I've created from the dye itself. Okay, now the reason why I say that, and to be fair, the ones that are in Inform are actually quite good, are really quite well developed. But with the tissue itself influences the spectral position. So if the tissue is old, for instance, say you're taking a block that might be 10, 15 years old, it's sitting there in the block, it's had some formalin still left in there because you just can't remove it. Over that time, it's become more acidic. Therefore, the pH will change, and the pH change will cause the dye spectral position to shift. And therefore, you know, it's not going to be perfectly the same as if you like a proposed spectral position. It also depends a little bit on the tissue itself, you know, whether it's the lung or whether it's liver or whether it's spleen. All these will have some effect on where the spectral position will shift to. So I personally prefer to measure my libraries from the sample itself.
Okay, thanks. Since we are almost close to our time, maybe we can take one more question here. Then for the others, we cannot answer here. Definitely, I'll send it to
you
Chris.
can send them to me.
Yeah, I'll say it later. The last question. What's your advice for the order of the staining prime antibody from more abundant protein or real proteins?
Yeah, so that's kind of an empirical decision to some extent. Now there is a list. Akoya has a list of where you should place the antibodies in terms of position, but the rule of thumb, as they say, is such that the dyes that are weaker in emission, you know, like the 690, like the more reddish dyes tend to be that in that area. You want to put your higher expression antibodies on there, right? And the reverse goes the other way. For the very bright dyes that you have, 480, 520, then these ones should be placed on the poor, the antibodies that are not so expressive. Also, it depends a little bit on where you place it as well. Now, as I saw in the slide, if I use Chi-67 as an example, it tends to be more productive if it's left towards the end of the cycles rather than the beginning of the cycle, where I think if you use CD20, for instance, it needs to be towards the beginning rather than the end. So you need to have a bit of an understanding, and again, there's references for this, is where how the epitope reacts to multiple cycles of antigen retrieval. And also related to that is where the best strength of the dye is going to be, you're going to use is concerned. So it all is a little bit of trial and error in this. So, you know, if people have it accurately documented, then that's a very good start. And I would certainly recommend that there's many publications now using Opal. So, you know, if you're contemplating doing an assay involving Opal, then please read up on what other people have done. They will usually give indications of where they've placed their components. So that at least gives you a helping hand. And then you have to kind of try it out yourself.
Okay, thanks. Thanks for all attendees, and especially thanks for Chris Johnson for today's presentation. Thank you. Yeah, I will collect all questions and pass it to you. Answer it offline. Okay, that's good. Okay, well, thank you everyone, and have a rest of a good day, and all the best with your multiplexing. Thank you very much. Thank you. Thank you.